And CD8 positive T cells induces a cell death of virus infected cells and tumor cells either by lysis or by apoptosis. So let's see how. Sorry, before that we'll try to zoom and understand what the T cell receptor complex is all about, so that we understand the functioning better. So the T cell receptor complex, so the T cell receptor complex is made of a heterodimer composed of disulfide linked alpha and the beta chains. We also have CD3, gamma, delta, epsilon, as well as theta chains, and together this is known as the T cell receptor complex. In addition to this, we also have other co-receptors such as CD28. This is with regard to CD4 T lymphocytes. The CD28 would bind to other receptors which are presented on the antigen presenting cell by the microbes. And this is a case of a CD4 lymphocytes which we have taken. So the CD4 receptor on the CD4 T lymphocyte would bind to the invariant region of the MHC class 2 here. The antigen presenting cell has processed the microbe. They are presenting the peptide on the MHC to the T cell receptor. The binding of alpha and beta occurs to this MHC peptide complex. CD4 binds to the MHC, and we have the first signal via CD3 theta chains, which goes intracellularly to tell the cell that we need to act or secrete cytokine. The second signal goes when the binding of the co-receptor CD28 occurs with the CD80 or the 86. Once both the signals are then transmitted intracellularly, we'll see what happens next. So here we have a CD8 positive T cells. There is the binding of the T cell receptor, which has occurred to the MHC peptide component. We have the CD8 here binding to the MHC class 1, which is present on the nucleated cell. So when the cell becomes infected with a virus or it becomes a tumor cell, there are the abnormal proteins which are generated. And it is these abnormal proteins which are presented by the MHC to the CD8 positive T cell. Once the signal 1 and the signal 2 reaches intracellular into the nucleus, there is transcription and then translation of proteins such as cytokines, granzymes, as well as induction of the death receptor signaling by the Fas ligand Fas connection. And once this happens, the cell undergoes apoptosis. So this is how tumor is normally dealt with in our body. So the question of why choose T lymphocyte, I hope it is answered, because these are the effector cells for cellular immunity. They constitute majority of the lymphocytes in the peripheral blood, thereby easier for collection. They play a major role in the immunosurveillance, potent anti-tumor agents, and they also help maintain therapeutic effect for many years after initial treatment due to the virtue of memory. They can also traffic to the site of antigen, and this is in particularly important with respect to solid tumors. So now let's understand what are the adoptive cell therapies which are based on T cells that exist as of now. So we have adoptive cell transfer using unmodified T cells. Initially, it began with tumor infiltrating lymphocytes, which many of you would know. So the principle here was that the tumor cells which have infiltrated the tumor, which you resect from a patient surgically, if you're able to extract those lymphocytes, you know for sure that they are able to intrude into the tumor, and they would have tried to attack the tumor antigen. They possess a very good anti-tumor activity against that particular tumor. So you extract those T lymphocytes and then you can reinfuse to the same patient. Then we come around the genetically modified T cells, wherein you can genetically engineer the T cell receptor. Basically, they genetically engineer the alpha and the beta to better deal with the tumor antigen or an antigen of interest. Then we have the chimeric antigen receptor. And also we have antibody therapies as of now, which is basically bispecific antibodies, which causes the T cell and it binds to both the T cell receptor as well as the tumor antigen, which you're going to target. And then we also have checkpoint inhibitors. So we'll concentrate on CAR because this class is all about chimeric antigen receptor T cell. So what are chimeric antigen receptor T cells? Here, what we do is we genetically modify autologous T cells as of now It's just autologous. So autologous T cells, in order to express a chimeric antigen receptor, which is specific for a tumor antigen. Once we have successfully expressed the chimeric antigen receptor XYO, we can expand the cells to reach the target dose and then reinfuse it to the patient. So this chimeric antigen receptor is basically a fusion protein. So what is this fusion between? We have a single chain variable fragment from a specific monoclonal antibody, which is targeted against our target of interest. Say, for example, the classic example here would be CD19. Now, if I need to target CD19, I would choose scFv of anti-CD19 monoclonal antibody. Now, the therapies in the market are mainly based on murine-derived monoclonal antibody, scFv region. So you take that as well as you combine it with one or more T cell receptor intracellular signaling domains. So it'll become better or clearer in the subsequent slides. Now, you know that the scFv, since it's an antibody, it's going to bind to the antigen, and that occurs in a non-MHC restricted manner. You don't require the MHC to process, to present it to the T cell for the T cell to recognize, because you have an antibody which already knows its function. So this is how a CAR looks like. Now, we have 4 generations of CAR as of now. The first generation of CAR, it contains, so this is the structure of the CAR initially. Firstly, we have an extracellular binding domain, which is derived from a single chain variable fragment. So we have the variable light and the variable heavy chain. The extracellular domain is connected to the transmembrane domain using a hinge domain or a spacer domain. The transmembrane domain would basically anchor this extracellular binding domain to the cell. And then we have the signaling domain. So if you see, in the first generation, they began with only CD3 CETA. So here, the problem that they did was, there's only signal 1 which is going intracellularly. So when they did the trial, they found out that the response is there, but it is not in the magnitude which is required. And also the response isn't persisting, beyond a certain point of time. So there was no clinical response for a good period of time. So then they came up with the second generation. So in the CAR has been divided into generations based on the number of co-stimulatory domains. So in the first generation, we have no co-stimulatory domain. In the second generation CAR, there is a co-stimulatory domain and a signaling domain. So all the therapies, the FDA approved therapies, are second generation CARs. Then you have the third generation, wherein there is more than one co-stimulatory domain. Then came the fourth generation. So in fourth generation, it's essentially the second generation CAR with an enhanced T cell function, such as a secretion of enzymes, cytokines, and co-stimulatory ligands. Now, this fourth generation CAR is also known as TRUC. That is T cells redirected for universal cytokine killing. And these are also known as armored CARs. So basically you have trucks and cars sort of, running around for the cancer immunotherapy. So let's try to understand here how is this whole, CAR expression, and what does a patient basically go through when the entire therapy happens. So the first step in the therapy is leukapheresis. Here the goal is that we'll need to obtain optimum range of CD3 positive cells. The range of CD3 positive cells from the clinical trials which has happened all over the world, anywhere ranges between 0.6 to 2 into 10 raised to 9 CD3 positive cells per bag. And this is typically collected from a non-mobilized donor, here being the patient, because it's only autologous CAR which is still approved. Once we do the leukapheresis, then the product is then sent for preparation of a CAR or manufacturing of a CAR production. The step 2 would be T cell activation or transduction, and then the cells are expanded in number once we reach the target dose that is calculated according to the body weight of the patient. So it ranges somewhere between 10 raised to 6 to 10 raised to 7 cells per kilogram body weight. Once we have achieved that particular number of CD3 positive cells, we can infuse the product into the patient. So since all of us or majority of us would be, very familiar with what leukapheresis what happens in leukapheresis. We'll try to understand the step 2 and the step 3 in detail. So just like it is applicable elsewhere, automation is definitely preferable, especially when it comes to CAR-T manufacturing. The only disadvantage with automation as of now is that most of the instruments, they're designed for a specific cell number and culture volume. So you cannot function beyond that range. Other than that, when it comes to labor requirements, training requirements, and a closed system processing, the consistency of outcomes, as well as the documentation, automation has an upper hand when it comes to manual. But it's just like doing an antibody titration on Neo. So without a transfusion medicine, say, resident or as a specialist, if you don't know how the manual technique is done, it's basically everything is done in a black box and you really don't know how it happens. So it is very important that you know how is CAR manufactured on a manual platform. So regarding the steps in the CAR-T production, it begins with first step being enrichment. So enrichment is isolation of the cell of interest. Here we would like T cells. So to get the T cells from this mixed bag of mononuclear cells which is present in the apheresis bag, that step is called enrichment. Now enrichment can be done in two ways. It can, there is density-based centrifugation or using Ficol, but the most commonly used method is the immunomagnetic cell separation. Now you have two techniques, either a negative selection or a positive selection. So in case of negative selection, you have the antibody, which is tagged to a paramagnetic material, and it targets the unwanted cells, which you would like to get, rid of. While in the positive selection, you have the antibody, which is targeting your cell of interest. So if I'd be doing CAR-T manufacturing or a CAR-T manufacturing, I would like CD4 positive and CD8 positive cells. So I'll have antibodies, anti-CD4 and anti-CD8, which I'd use for my positive selection. So in a manual platform, you can perform either a negative or a positive selection, but in the automated platform, the CliniMax Prodigy, typically it's a positive selection which is done. So it comes with its own advantage. So negative selection, your cell of interest is basically untouched and kind of naive for you to work on. While in a positive selection, the advantage is that you will have the pure, just pure CD4 and CD8 positive cells without any other contamination. So this is how it is done. Now you begin with an heterogeneous population of cell in suspension, and then the cells can be labeled with your antibody of choice. So it is basically an antibody which is, tagged to a paramagnetic material, and after which you subject it to a magnetic field. Once the magnetic field is applied, in case of a negative cell selection, all your unwanted cell is going to settle down because those were tagged with the antibodies, and supernatant is what you really need. So you take the supernatant and then resuspend it in the media of your choice. While in positive selection, your cells would be settling at the bottom and you can discard the supernatant. So once your enrichment is done, you'll go into activation. So what is activation? So in vivo, the naive T cells, they are stimulated for proliferation and differentiation by antigen presenting cells such as dendritic cells. Now the activation happens by the interaction between the MHC and the T cell receptor. So the naive goes on to become a stem cell memory, and then central memory, effector memory, and then effector cell. Now if you see, when this activation happens, the memory function sort of deteriorates and the cell becomes more of an effector phenotype. Now, in vitro, how do I do this? So this is in vivo that they've shown. You have the antigen presenting cell, and then you have the MHC, which basically binds to the TCR, and the co-receptor CD28, which binds to, say, CD80 or CD86. The cell becomes activated, and there is secretion of interferon gamma and TNF alpha. Now, in vitro, how do I do this? There are three platforms as of now. You can use monoclonal antibodies and interleukins. That is, your target would be CD3. So you can use anti-CD3 and anti-CD28 monoclonal antibody along with interleukins. There are also cell-sized beads coated with anti-CD3 and anti-CD8 along with interleukins. And also there are artificial antigen presenting T cells. Now, why do we need to do activation is because for our next step of transduction or transfection, this would sort of set the background. You know, it'll prepare the cell better to receive the virus. So the step 3 would be transduction or transfection. So there's always a confusion of this term. Transduction is when I'm going to insert the gene of my interest using a viral vector. That is called transduction. When I'm using non-viral methods, it is called as transfection. So here, this was a beautiful picture that I found, wherein this is a virus and you're trying to insert your gene of interest using the virus into the T cell. So it doesn't happen like this. The virus doesn't go in. It's just that it fuses with the cell membrane and the gene of our interest goes in and integrates with the host genome. So when it comes to the gene delivery systems in CAR, broadly you can divide it into viral methods, mostly based on lentivirus and retrovirus. There's more use of a lentiviral transduction, which is used in CAR-T than a retrovirus. We have non-viral methods such as electroporation, wherein a high pulsed voltage is given to the cell. There are, transient pores which are made and the gene of interest just gets in. We also have cell squeezing as well as nanoparticle based, but it is not really used. It is mostly either electroporation or you have viral methods which are used in CAR-T. Now, this is very interesting, and I'm hoping that this would be something new on how a lentivirus particle is produced. So what we do is, we take a packaging cell. The packaging cell usually taken is HEC293, that is human embryonic kidney 293T. It's a cell line. You have it in a plate, and then you can add the plasmids which contain your gene of interest as well as other genes which is required. So as all of you would know, lentivirus is a retroviridae. Now, from the retroviral genome, I would like to choose genes which are only and only essential for the viral particle to form, such that the gene of interest can be delivered to the patient's T cell. So we have for the packaging plasmids as well as the transfer plasmid, which you'll be adding to your packaging cell. So this packaging cell is going to act like a machinery or a virus factory, wherein all the components would come together and you can harvest the viral particles. So here you have the GAG and the POL gene in the first plasmid. So the GAG gene is required for formation of this capsid core, which holds the gene of interest. So if you're able to see it properly, so there's this blue colored ring. So that's going to be the capsid core which holds the gene of interest. So if you need the capsid core, you need the GAG gene. Second is POL. Now POL is very crucial because for the reverse transcriptase enzyme, because the gene of interest here is going to be RNA, you will send it to your T cell. You want the RNA to be converted to DNA so that it is able to integrate into the host genome. So for the reverse transcriptase, you need POL. You need POL also for integrase, which will help the DNA to integrate into the host genome. So I have got these two genes. Now you have REV. REV will help in. So once the gene of interest has integrated into the host genome, the DNA would, transcription happens, the mRNA is produced. Now the mRNA needs to reach from the nucleus to the cytoplasm. So REV would help in that. Only once it reaches the cytoplasm can the protein be made. Now the third is called the envelope plasmid. So if you remember correctly, HIV has got ENV gene for the envelope. However, you have here we have changed it. It is VSVG. It is called vesicular stomatitis virus G. Why? This is called pseudotyping of the lentivirus. So what we are trying to do here is that in order to broaden the tropism, so you don't really want the virus to only target the CD4 positive T cell, which by ENV gene that it has, you would like to broaden the tropism. For that, we have the VSVG. And also it would, help in the virus not really turning into an infectious particle. So you have these three plasmids, which are called the packaging plasmids, and then you have the transfer plasmid, in which you can insert your gene of interest. So this would basically contain the amino acid sequence, which would form the CAR sequence. So the SEFE, the hinge region, the transmembrane, as well as the co-stimulatory and the signaling domain. So that gene would be inserted here. Once I put this all into the packaging plasmid, so this is a very beautiful diagram. Here you have the gene of interest. This is the RNA. Then you have the GAT-POL and the REF, as well as the VSPG. Now the viral particle is generated. So the packaging cell is usually kept in these plates. They would release all the virus onto the supernatant, and we take the supernatant. So that is where the lentiviral particle would be there. Then we estimate the titer, and then we use it for transduction. So this is the patient's T cell. So here the virus comes and fuses. There is the capsid is sort of dissolved. We have the RNA here. RNA gets converted to DNA because we have POL, which will give us the reverse transcriptase. DNA will go with the help of integrase, go and integrate into the host genome. And from the host genome, it comes out with the help of REV, which will bring out the mRNA out. And then we have the recombinant protein, which is CAR. So once we have done the transduction, now we would want the cell to expand in number so that we have a good optimal dose of T cells, CAR T cells, which we can give to the patient. Now the T cells, so here the thing is we need to maintain a ratio between the culture, the media and the T cells. So for that, we have a number of vessels in which this is essentially done. Classically, the T cell culture is done in a T flask. So in this diagram, what you see is a T flask. And these are all the media. There's phenol red, and that's the reason why it's all red. And you have the serological pipette here. We can add the media, add the substrates which are required. That's how all these steps are subsequently done. When the cell number increases, say usually we maintain it at 1 to 2 million cells per ml. If the number goes beyond that, I'll need to have a bigger vessel. So T flasks are not really the go-to choice when you need to manufacture it for a patient. For a research and development setting, T flask would be good. So we also have other methods such as a T cell culture bags. Now these T cell culture bag, it is made of fluorinated ethylene propylene. It's called FEP. It doesn't bleach. It doesn't leach, and it's chemically and biologically inert. Now I can add the media and the interleukins and whatever is required subsequently for the culture by using in a sterile condition, because it's just like your blood bank. You can connect it using an SCD or a sterile connecting device. So this is another good choice that we have. And most of the products which are there in the market out now is produced using this. This is called a rock motion bioreactor or a wave bioreactor. So what happens here is that we have this multi-layer plastic bags, which is mounted onto an adjustable rocking platform, which you can see here. It's similar to our platelet agitator, we can say, in a way, because using these tubes, we can add the media, the cells, the interleukins, and whatever is required. And there's this rocking wave-like motion which happens. The bags being gas permeable, the cells can respire and increase in number. So this slide, it tells us that what are the current CAR-T manufacturing routes that we use, sort of combining all what I'd said as of now. So the activation, the majority of the clinical trials as well as the products use anti-CD3 and anti-CD28 beads. Gene delivery, commonly or majority of the people do it via lentiviral transduction. Expansion is done using a rock motion bioreactor by majority of the companies. And so the platform essentially is a bead-based activation with a retroviral or a lentiviral transduction and expansion in a rock motion bioreactor. And these were all the manual steps. Now, this is the automated platform which is available as of now. It's called Clinimax Prodigy. So here they've got a 12 to 14 days TCT or a T cell transduction program. This is basically a black box or a machine which will help us do all the labor intensive tasks, which, manually otherwise we'd have to do outside. So we have completed our step 2 and step 3 as well. Now your cell is ready for infusion. But prior to infusion, you'll have to prepare the patient. So we give a lympho-depleting chemo, usually a combination of fludarabine and cyclophosphamide. So this is done so that we can, set a good niche for the CAR T cells to go and establish as well as, perform the function. And also the tumor burden would decrease. And that's very important for a CAR T. I would tell you why. Now, once I've infused the product, what do I expect the CAR to do? So here you have the CAR. This is the SCFE region. It goes and it meets the tumor antigen. So we'll consider it as, CD19. This would be the B cell. This is the tumor antigen CD19 that we're targeting. This is our T cell, the patient's T cell. The CAR would bind to the tumor antigen. Once this binding occurs, there are two signals. There's an intracellular signaling cascade which is activated. The first signal goes from the CD3 zeta via phosphorylation mechanisms. Then you have the co-stimulatory domain giving the second signal. Once this is done, the transcription of T cell effector function happens by release of interleukins. The cell proliferates, and also there is generation of perforins and granzymes. These would cause pores in the tumor cell, resulting in a cell lysis. Now let's see what are the FDA-approved CAR Ts in market. So there are 5 FDA-approved CAR T therapies in market as of now. Majority of the products target CD19. We have one product for relapsed refractory multiple myeloma targeting BCMA, or the B cell maturation antigen. So these are all going to be a little difficult to pronounce words. You have tisagenlecleucel, or the brand name being Kymriah. It is by the company Novartis, the first to bring CAR T into market. We also have axicabtagene ciloleucel, brexucabtagene autoleucel, idacabtagene vicleucel, and lisocabtagene maraleucel. So these are the FDA-approved CAR T therapies in market. Now, it's not just essential to say that there's a therapy which exists and this is how. Ultimately, the question would be, how is the clinical response? So with respect to Kymriah, they have found 82% of the people with overall response rate. That is, overall response essentially entails complete response plus partial response. We have the Yescarta having 44% of the patients alive at 4 years. And here the thing is, we are treating relapsed refractory patients whom we don't really expect to, survive more than say 3 to 6 months, because they have really exhausted their options of therapy. So giving them a 4-year chance is something I think is incredible. Then we also have Abecma, which has a 72% response, Breyanzi, which has got a 73% response, and Tecartus with 87% overall response. And just like all the other therapies, this therapy has its own complications. And the classical or the hallmark clinical toxicities, which is associated with CAR T therapy, is cytokine release syndrome as well as neurotoxicity. And the strange part was that it was not predicted by animal modeling. So you begin with a preclinical, and then only then you come to a phase 1 or a human first in human trial. So I will also tell you why. In CRS, what happens is it typically manifests within the first week with signs and symptoms of fever, hypotension, respiratory insufficiency, and a high serum cytokine level, classically a very high IL-6 level. The patient can also have neurotoxicity characterized by a temporary working memory loss, delirium, seizures, and rarely acute cerebral edema. Now, the attributable etiology for both is rapid activation and expansion of T cells, which secrete cytokines. As far as CRS is concerned, what actually happens is that there are cytokines which is released because of the CAR T and the tumor antigen interaction. Once that happens, there is a small amount of cytokine which is released, which recruits the rest of the cells to come into picture. Now, these cells, they also have their own cytokine secretion, and these are the immune cells of the myeloid compartment. These cytokines further aggravated by the naturally occurring adrenaline in the patient becomes basically a vicious loop of cytokine storm. Now, why did the scientists and the clinicians, why were they not able to predict it in the first in human use? Was because the preclinical model which is used is not SCID mice or the NSG mice, and which lacks the myeloid compartment. So that is the reason why they were not able to predict such a side effect before they used it in a human. And this is the mechanism of neurotoxicity. So once all these pro-inflammatory cytokines are there within the blood vessel, this is the blood-brain barrier which is depicted here. Now these pro-inflammatory cytokines, they would activate the endothelial cells. When the endothelial cells are activated, there's increased permeability as well as capillary leak, causing a vasogenic edema. This would result in a focal edema and neurotoxicity. Now, so we have understood what are the side effects which are associated with the therapy. And tumor cells, you know, they are these intelligent beings, I would say, that they have even found out how to resist this therapy. So there are basically two mechanisms of resistance. One is antigen-dependent resistance, and we have an antigen-independent resistance. So in antigen-dependent resistance, how has the tumor cell kind of thought? So the tumor cell has understood that it is CD19 that, you know, these CAR-T is directing. So why not cause a point mutation or the loss of gene? So you have these B cell leukemic or the lymphoma tumor cells which are running around in the patient's body without a CD19. So basically the CAR-T is blind because it's not able to detect the target. The second is wherein, so this is a very rare case which occurred, which I read in one of the papers, wherein I'm assuming that the isolation that they would have done is probably a negative isolation. So what has happened here is just prior to the transduction, so the whole collection contained both the T cells as well as a tumor cell. Now this tumor cell, say the B lymphocyte here, has CD19 and it has got transduced as well, because if you remember, we have done the pseudotyping of the lentivirus. It has a broad tropism. It will go and infect whatever cell that is there, right there in the front. So the tumor cell has also got affected. Now what happens is the same cell has got the CAR as well as the CD19, and the CD19 is essentially masked. Now what happens, the tumor cell knows that the other tumor cell in the patient's body, when you infuse the product, is going to get killed because the CAR would interact. But this particular tumor clone will survive. Now they'll try to preferentially have the progeny from this tumor cell, which would have both the CAR as well as the CD19 antigen, thereby escaping the mechanism. Now the third mechanism is called trogocytosis. What is trogocytosis? The CAR basically binds to the tumor antigen and plucks it out and then redisplays it on the T cell surface. So when there's decreased antigen expression on the tumor cell, when that happens, obviously these guys can actually escape the CAR-T from attacking them. Then we have antigen-independent resistance. So wherein if you see, the T cell culturing is mostly restricted for say 12 to 14 days. We wouldn't want to go beyond, even if that is going to help us increase the number or expand the T cells in fashion. The reason is because if you would remember the slide on activation, the knife upon activation finally becomes the effector T cell. I really don't want the effector phenotype T cell in the product. I would prefer having more of central memory and stem cell memory, because it has the right balance of both the memory function as well as the effector function. Now, if you are gonna give the patient more of an effector phenotype like infusion product, it is basically like sending old men or women to the army battlefield. They are going to get exhausted very fast. The second mechanism is wherein you remember that there's this Fas ligand and the Fas which comes into picture, and the death receptor gets activated and the cell has to go into apoptosis. Now, if the death receptor signaling itself is lost, the tumor cell can survive. And the last and the third mechanism is the immunosuppressive tumor microenvironment, much more relevant in case of a solid tumor. So here what happens is the tumor kinds of protects its environment by secretion of all these cytokines that the T cell is unable to infiltrate into this immunosuppressive microenvironment. So these are the ways in which the tumor cell has developed resistance to this therapy. Now, we have, so there are multiple trials which are going on in solid tumors as well, apart from, the leukemia, lymphoma, which is become sort of the classical, and it is kind of become saturated as well. So The country to lead the trials in CAR-T in solid tumor is China, followed by US. And India is yet to kind of open its account, but I'm hopeful that in, say, the coming 3 to 5 years, we should also be there somewhere on the map. And they have targeted almost all the solid tumors which is possible, with most of the therapies targeting brain, CNS, tumors, glioblastoma. There's been incredible research on that happening. We also have trials on lung, breast, as well as pancreas. But there are a lot of challenges with solid tumors. If you'd see, there's none literally which is an approved CAR-T in solid tumor in the market because of the various challenges that they're facing, such as the potency of the living drug causes a lethal on-target toxicity if it is expressed in life-sustaining tissue. So here, if they have, say, adenocarcinoma colon is targeted and you target CEA, it is also expressed in the other normal colon cells, and the patient did result in severe colitis. So when it is present on a life-sustaining tissue, there's lethal on-target toxicity. So the scientists are kind of caught in a situation where they have to find a uniquely expressing tumor antigen only on tumor cells and not on normal cells. Then the active proliferation of the CAR-T, this is common for both the hemat cancers as well as solid tumors, that they have serious side effects such as CRS and ICANS. And then there's insufficient sustenance of response because of the resistance to the antigen loss, and as well as the immunosuppressive microenvironment, which I've already told. Now we've understood that the CAR-T in hematological malignancies is there. We have seen it in solid tumors. So what next? Now, these are the other eligible, targets out there. NK cell, macrophage, and Treg, of which NK cell is the most kind of, eligible bachelor out there, because NK cell already has it's a part of innate immunity. It has got a native antiviral and anti-tumor effect that it has got, and we can really use that to target the tumor cells without much of cytokine release, because therefore it offers better safety when compared to a CAR-T. And also it has got multiple mechanisms to activate the cytotoxicity, unlike a cytotoxic T cell. And they also have a high potential as an off-the-shelf therapy. So especially in solid tumors, CAR-NK, the number of trials are really high. We also have CAR macrophages. So here, what happens is when the signaling happens, the macrophage sort of shifts into an M1-like phenotype. M1-like phenotype is a pro-inflammatory phenotype. And this would cause an CAR-induced phagocytosis of the infected cell. We also have Treg, and as all of us would know, Tregs helps in preventing an allograft rejection and also autoimmunity. Now here what we are doing is essentially trying to boost its function. So this is going to be very relevant in a transplant setting as well as autoimmune diseases. So these are the a few trials which have happened over the course of years. Since the first FDA approved therapy. So we have CAR transfused NK cells in CD19 positive lymphoid tumors. We have, tried to engineer the CAR-T to navigate the immunosuppressive microenvironment in a solid tumor. We've also had dual CARs. So you know that if you target one antigen, the tumor is gonna, come up with this mechanism of resistance. So we target two antigens. The most common is CD19 and CD22, which are targets. And then this was a very interesting, in fact, research, which I came across. I think this was in August 2021, that in NEGM, there was a correspondence of CD19 targeted CAR-T cells in refractory SLE. The basis was that CD19 would target the B cells, and the B cells, if destroyed, the patient essentially goes into a B cell aplasia. So you don't have any antibodies, which the patient is going to produce, thereby no autoimmunity. So the patient being refractory, she had undergone, I think, almost all the standard therapies for SLE and yet didn't respond. She was given lymphodepletion using fludarabine, and then CAR-T was given and she has gone into remission. So that was something very interesting. And what the CAR-T people in the CAR-T domain look up to as the future of CAR is Allogeneic CAR, because as doctors we really know that it's much easier to have something off the shelf. Take a prescription pad, just write it down so the patient can get an infusion in the OPD basis and just go home. So that is not possible for an autologous CAR because of all the labor intensive steps which we have to do, and essentially because of the GVHT that we have to tackle. So obviously they've come up with the mechanisms to tackle that. So let's see. The difference between autologous and allogenic CAR. Allogenic CAR, I really hope that you clear with the steps. We have the leukapheresis, the cells are proliferated. We do a T cell transduction using a lentiviral vector. The cell becomes CAR positive. We expand it, cryopreserve it, and infuse it to the patient. While in an allogenic CAR, what is different is that once the CAR is expressed, what you can do is electroporate the cell, cause a knockout of the T cell receptor and CD52, which is essentially responsible for GVHT, expand the cells, cryopreserve it, and then the leukapheresis from one healthy donor can help a lot of patients. So that is what allogenic CAR is all about. So here, as you can see, this T cell, that is an allogenic T cell, if I need to give a host, if I need to administer it to a patient without a HLA match, just get rid of the TCR, which is responsible for causing the GVHT, and there's no more recognition of the foreign antigen that happens, and then you are free to administer it to any patient of your choice. So that was the basics and recent advances in CAR, and I would like to conclude the discussion with few points. So CAR T is a form of cancer immunotherapy involving genetic modification of T cells. The steps which are involved in CAR manufacturing are enrichment or isolation of cell of interest, after which you activate or stimulate it, following which you can transduce it or transfect it, expand it, and then administer it to patients. There are 5 FDA-approved CAR T therapies in market as of now. The most common clinical toxicity is cytokine release syndrome, followed by ICANS or the neurotoxicity. And the other upcoming CARs are CAR-NK, dual CAR, allogenic CAR, or universal CAR. So this was one of the first NEGM perspectives which had come out on the tragedy, perseverance, and chance, the story of CAR T. I've always felt that they should have added a word called hope as well, because we are essentially giving hope to these relapsed refractory patients who have exhausted their options. So here we have Carl June, the person who has brought CAR T a reality, and she's the first patient who was treated with CAR T, Emily Whitehead. So thank you all, and we can take up any questions if there are any.